timeline edition because there's several different timelines that we'll be looking at. So the things that we will be discussing this session is the viral and bacterial evolution studies. There's been a number of those that have come out recently. We're going to look at a few ancient microbial isolates and what the microbes of the past can tell us about the future. We'll talk about prion denaturing and some protein folding artificial intelligence. And then finally, we'll end with a new report looking at how many multi-drug resistant related deaths there actually are. Let's dive right in. Our first set of studies comes from Nature Communications and ASM's MBio journal. The Nature Communications study is titled High Throughput ANI Analysis of 90K Prokaryotic Genomes Reveals Clear Species Boundaries. And the MBio study is titled Origins and Evolution of the Global RNA Virome. So the take-home message of these two different studies are basically that microbial species, yes, and microbial evolution, getting there. In terms of whether microbial species do exist, this study fairly well establishes that they do have separate and distinct species, and we'll get into what that means. But the role of evolution, of course, is something that is ongoing, and understanding how microbes have evolved is something that will always be changing as we gather more information. So the first study here from Nature Communications looks at the question of whether microbial species exist. There are basically two different ways you can think of microbial species. One is that there are distinct clusters, that is, species that cannot interact with other clusters. And it's kind of hard to study microbial species the same way that we study, say, metazoan or animal or plant species, because in animals and plants, we look for whether two adults can have viable offspring. So whether their offspring can have offspring. And of course, sexual mating doesn't happen in microbes. We can look at horizontal gene transfer and whether that's possible as one potential marker of speciation. But the alternative explanation for clusters would be a gradation or almost a continuum of diversity, looking at just a somewhat arbitrary cutoff of different number of nucleotides as setting that as this is a different species. And so I'm showing you a graph from, I think this was 2005 Science Commentary, looking at the 16S ribosomal RNA gene sequence, which is how species were determined for a long time, looking at a 90 or 95% identity to count two microbes as the same species. Here you can see that it's somewhat of a gradation looking at the number of different nucleotides that can be found between different, here we have genus demarcations, species, and then strain would be the most similar. Now, understanding whether these species exist is important not only for an academic exercise and collecting the number of different diverse organisms on this earth, but also has practical consequences with regards to things like diagnosis in clinical microbiology laboratories, whether microbes can be transported across different geographical or country boundaries, and whether those microbes, if they are discovered, need to be quarantined, for example. And so now that we are able to do whole genome sequencing at a much more efficient rate, one of the ways to determine relatedness is to look at the average nucleotide identity, or the ANI, between two different genomes, often depicted in this graph type that I'm showing you on the right-hand side, where two different genomes will be compared. And then their similarity in this case is colored either green for similar, dark green is the most similar, then shades of neutral going into red as dissimilar. And so you can see, of course, if we have basically the same list of microbes going down, they are all related to themselves, dark green, as one would expect. And you can see in this case, certain clusters of green where the different genomes have more nucleotides in common across the entire genome, not only looking at a single gene. Now, looking at 90,000 different species, 90,000 different genomes, which are deposited within different registries, would be able to determine whether or not all of these different genomes cluster in various clusters, as we see here, or if there's a continuum across all of those different species, or all those different genomes, excuse me. And so this group developed a new algorithm that would be with high computing power. Of course, this sequencing comparison is really becoming a computing problem as much as it is a microbial evolution problem. And so this new algorithm had to be verified, which they did. And then they looked at all of those different genomes that had been deposited within the NCBI genome database, 91,761 prokaryotic assemblies. And you can see right away that there are distinct clusters. We have here above 96% identity, a cluster of different types of, here we're looking at organisms at the genus level, Escherichia, Salmonella, Myxococcus, Pseudomonas, etc. And then less related organisms start to cluster again with a large gap between in the upper 80s and lower 90s, things that have say 75 to 80% identity. So this supports that there are actually distinct species and that it's not a gradation of just a few nucleotide differences that differentiate different microbial types. And so in following up this different information, the researchers wanted to look at whether genomes deposited at different times might give different results. And that's because at first, when genomes were being accumulated and sequenced and deposited into the genome databases, researchers were looking to just get a heterogeneous mix of what types of microbial diversity was out there. And so those were expected to be more different because if you're testing, say, different samples of human and plant and soil and all the different niches in which microbes reside, those microbes would, of course, be very different from one another or expected to be very different from one another. But as whole genome sequencing has become more efficient and people are interested in strain to strain variation, more related the recent sequences have been more related to one another, as one would expect. And so they looked at various generations, you could say, of these genome deposits. First 1,000, the first 5,000, the first 25,000, and all of them. You can see that regardless of when those genomes were deposited, that there is a very distinct gap between those that have the same identity and those that are somewhat related, but only 75 to 80% ish. And they did that looking at same species, different species. You could see E. coli and Shigella actually group with what they would call the same species. We know that those are very related organisms, differing only in a few toxin genes in some senses. And so that, oops, sorry, we're going to transition into looking at the virome. And so that in this case, the researchers were looking at the majority of RNA viruses. And they wanted to look at RNA viruses and RNA genomes because the majority of diverse viruses that infect eukaryotes that are either plant pathogens or animal pathogens have RNA genomes. Now, of course, up until recently, virology has really focused on those pathogenic viruses because they have a very clear phenotype and are much easier to identify. But looking at the different viral diversity using metagenomics and just looking at what sequences are out there without a bias has increased the RNA virus identification capabilities. And some of these new identities have reorganized RNA viral lineages. And so the researchers wanted to see if they could remap out some of the lineages, looking at how viruses have evolved over eons. And to do this, they were going to look at a few of the different viral genes that are required for RNA to be both expressed and copied within the eukaryotic host. So if a virus has an RNA genome, it needs to make more copies of that RNA genome. And to do that, it can use a number of different enzyme complexes. Can use something called an RNA-dependent RNA polymerase, where it will copy the RNA directly using a viral enzyme or an enzyme complex. It can use a reverse transcriptase, which will convert that RNA into a cDNA, and that cDNA can then be used as a template either for genome copying or for generating different types of mRNA or positively stranded RNA that the cell machinery can then use to make the different other additional viral products. And of course, all of this depends on whether or not you have a positive sense RNA virus or a negative sense RNA virus, which is basically saying positive RNA, that's the type that the eukaryotic cells make already. They can use it directly to make mRNA, or those that are negatively sensed, which then has to be converted into a positive strand RNA. And various combinations of these different enzymes are required for different types of viruses. So the group took a reconstruction of an RNA virus evolution, and they used this RNA-dependent RNA polymerase tree as a scaffold. And so they took these 5 different branches of viruses, as they showed in their paper, the tree as it was understood. And from here, they were able to make a lovely schematic of the transition of viruses from their initial prokaryotic hosts into eukaryotic hosts. And one of the things that I really like about this schematic is that it shows the viral evolution, which you can see the last common ancestor of the major branches of the positively sensed RNA viruses only encoded this RNA-dependent RNA polymerase, and they also predicted a single capsid protein, which then branched off into the different lineages here. Excuse me, but this evolution was occurring, of course, in tandem with other types of evolution, including the emergence of eukaryotes and more complex life forms to which these different RNA viruses were able to adapt. And so these are very exciting studies because they look at some very large data sets in order to answer some of the questions about how life formed on Earth, how diversity occurs, what does diversity look like, whether it is a continuum or very distinct organisms. But this was not really picked up by a number of mainstream articles or mainstream news outlets. It was discussed quite a bit on social media with people discussing about whether microbial species are actually a thing, and other scientists saying, spoiler, microbial species are a thing. Of course, it's nice to have 90,000 genomes interrogated in order to support that hypothesis. And the MBio piece was talked about and discussed and excited scientists even before it was an MBio piece. It was published originally on bioRxiv, a repository for preprints that are the studies before they've been under peer review. And people have been looking forward to seeing the final form of this study for quite a while. Excuse me. All right, we're going to move on to our second story. And this is, I have 2 different research papers highlighted here. We're going to focus a little more on the cell paper. I have another one from the International Society for Microbial Ecology, and we'll discuss that briefly at the end. And so basically, this is going to talk about a plague bacterium that existed in and shaped Neolithic life. So this is the summary from the cell paper titled Emergence and Spread of Basal Lineages of Yersinia pestis During the Neolithic Decline, with the second paper from ISME is Resuscitation of Anammox bacteria after over 10,000 years of dormancy. So the Neolithic era is the Stone Age, the late Stone Age, when mega settlements of people, meaning maybe 10 to 20,000 humans, started settling in things that were more similar to the cities that we know today, in places mostly around Eastern Europe. I think Romania, Ukraine. These settlements were relatively short-lived, maybe a couple hundred years, and were often abandoned and even burned, with the charred remains being discovered by archaeologists. Settlements were not from this Neolithic era were not discovered after around 5,400 years before our present time right now. And this is thought to correlate to population decline around this time, which is a mystery why this population decline occurred. It could have been environmental overexploitation, using up all of the natural resources nearby, as humans and lots of other organisms tend to do. But it also may have had an infectious disease at its root. So I'm showing you here a picture of some stone, I don't even know, settlements or stone figures from Sweden. So this, I wanted to find something from the Neolithic era of Sweden because the research here focuses on a Swedish grave site from about 5,000 years ago, looking at the genomic information that people were able to recover from some of the samples there, scientists discovered that there was bacterial sequence similar to Yersinia pestis. And this was a big surprise because plague was not known in the Neolithic era. People have, up to this point, considered Yersinia pestis to have emerged from probably the Asian steppes a couple thousand years ago, not 5,000 years ago. We've discussed even some of those studies. And so what the researchers did was, of course, to get the sequence of this strain, which they called GOK2 or GOK2. And by looking at the different nucleotides sequences and comparing those to modern day and other ancient lineages that are not quite that old, they were able to determine that this GOK2 lineage emerged and spread during the Neolithic era. And so they wanted to not only look at how GOK2 was able to relate to some of the other lineages, as you can see here, kind of a timeline of evolution of Yersinia pestis, but they wanted to compare how this sequence may have interacted with ancient human genomic sequences. Um, and so they could predict perhaps both how this bacterium was able to interact with different human populations that were spread throughout Europe, and perhaps look at how it was able to, how these different human populations were able to lead to new lineages or new places where the Yersinia pestis may become endogenous. And so they were able to make this lovely map schematic. Reminds me a little bit of the one that I just showed you, another timeline showing, in this case, not the evolution of complex life, but other things that have happened in relatively recent human history in the last, 5 to 10,000 years here. And what they found is that the plague pandemic was likely to have emerged earlier than expected and spread throughout these large settlements, and perhaps been passed between these large settlements, which of course were relatively sparsely populated in the in between areas. So it wouldn't create a reservoir necessarily in the in between areas, but perhaps become established within the rodent and flea population within those mega. Settlements and spread over the trade routes between that existed between these mega settlements, eventually making it over to the Asian steppes, which is where we've talked about the emergence of the Yersinia pestis strains that caused the Justinian plague, that caused the Black Plague in Europe, and other major Yersinia pestis epidemics. And so this was written up because, of course, anything that rewrites human history could be quite interesting. And the speculative nature of the spread of the Yersinia pestis is discussed in this really nice piece in the Atlantic. Of all the pieces that I'm going to highlight today, I very strongly endorse this piece in the Atlantic by Sarah Zhang. I thought it was very well written, and she um, was able to get a quote from an archaeologist who says that this is they've got some really neat DNA results, and they're building a story about it, and it's an interesting and potentially plausible story, demonstrating a little bit of skepticism or at least requirement for additional DNA sequences. And finding some additional evidence may be a little bit difficult since those mega settlements, despite being hubs of human activity, don't have associated grave sites. And what these Neolithic era humans did with their dead is relatively unknown. So finding more Yersinia pestis samples that are associated with the remains of people that lived at that time might be a little bit difficult. And it was also shared in other outlets such as Science News. Quite exciting. But this is not the only ancient bacterium that has been studied recently. I want to quickly highlight this ISME paper because what they show is that an Anammox bacterium can be revived simply with water, not with any additional external stimuli. No additional nutrients are necessary. So these Anammox, which are anaerobic ammonium oxidative bacteria, were resuscitated from a dry terrestrial ecosystem after resting for over 10,000 years. That's quite a long time to be metabolically inert. Lots of microbes can become metabolically inert given desiccation, such as a dry condition or other types of external stimuli, but showing that they can revive with minimal stimulation is something that is quite interesting in this case. And on the right-hand side, I'm showing a few different ways that the um, that ammonium oxidation can occur. They were able to do isotope testing in order to determine that these different microbes were at least 10,000 years old. And they showed that these revived microbes reduced nitrate to ammonium, which would provide the substrate ammonium for the anammox. And then this anammox, or I'm sorry, the ammonium plus the nitrate formed were used to produce nitrogen gas. And the reason that this is important is because these anammox bacteria play a large role in the global nitrogen cycles. And so, as global warming brings precipitation to areas that are not necessarily receiving a lot of water at this point, this precipitation may revive dormant bacteria in soils where they are metabolically inert and have impact or a great effect on the global nitrogen and potentially carbon cycles, depending on what the substrates used are. And so this is a very interesting and perhaps foreboding study showing that some of the ways that we know the global nitrogen and carbon cycles to occur may undergo large changes in the near future. All right, moving on, we're next going to discuss a paper from PLOS Pathogens. This is a paper titled Soil Humic Acids Degrade CWD Prions and Reduce Infectivity. The take home message from this study is that soil components degrade hard to denature prion proteins. This is a pretty big deal because prions are very, very stable. So let's back up and talk about prions. They are infectious proteins, which, when discovered, was a big controversy within microbiology and infectious disease because the prion itself has no genetic material. There is no RNA, there is no DNA, and this went totally against the dogma of what an infectious particle was. This is such a paradigm shifting discovery that a protein could actually cause a disease that it won the Nobel Prize in the 90s for Stanley Prusiner, who did a lot of work on prions. But the way that the protein can act as an infectious particle is that it is a misfolded protein. So there, in this case, on the right hand side, I'm showing you a normal protein. This is a prion protein called the conventional or normal protein, which can become misshapen. And this original misshapenness or misfolded protein, how this occurred is not well established, but it becomes a prion protein that is the scrapie form, in this case, of this particular prion protein, called scrapie because this was studied heavily in sheep in a disease called scrapie. And those scrapie form of the prion proteins are able to convert the normally folded proteins into the prion, the scrapie form. And so this is something that will, once there is one within a system, it can be a cascading effect as the misfolded proteins are then able to induce refolding within the normal proteins in the whatever organism it is infecting. This normally happens in the central nervous system, and so it will eventually cause protein aggregates and occlusions within the central nervous system, which is, of course, includes things like brain tissue and can have a lot of deleterious effects. Now, these prions, the scrapie prions, are extremely stable. So that means that they do not, based on being dried out or desiccated or being exposed to certain harsh chemicals, they don't change their shape. And so that makes them potentially dangerous if they're able to hang around and become ingested and somehow make their way into the central nervous system of another normal, healthy organism. And that's what happens with chronic wasting disease. So the CWD is chronic wasting disease. It's a disease of cervids, which are things like deer, moose, elk, and it is basically these prion proteins that will cause a degenerative central nervous system disease. I'm showing you some brain tissue here from, I think it's reindeer on top and moose on the bottom, I believe, with the aggregations of proteins stained with antibodies here. So you can see it can cause quite a lot of protein aggregation. And as those animals succumb to the disease and die, they will not die on a sterile surface. Of course, they succumb within a forest, and you can see that they will eventually decompose and release those potentially infective prion proteins into the soil and become part of the ecosystem there, and potentially affect another deer if say this deer I'm showing decomposes the it's fall here, there's a lot of leaves, but eventually there will be new grassland, and that grass could be contaminated with some of the prion proteins from this deer that has died from chronic wasting disease. This group wanted to investigate whether the soil composition could affect prion stability and infectivity, and particularly they wanted to look at humic acid. And the humus, as the top part of the soil, contains a number of different types of organic compounds, humic acid being one of them. This is a group of compounds that have a molecular weight that can vary from 10 to 103 kilodaltons. It's insoluble under acidic conditions, and the different types of humic compounds can vary among soil types across the world. You can have just a tenth of a percent of humic acid in that top rich part of the soil. You can have up to 25 or more percent. And so what they did was to expose some of these prion proteins to various amounts of humic acid and determined that this soil organic matter does degrade these chronic wasting disease prions. You can see an increasing effect of humic acid. There is less and less of this prion protein that is still stained by antibody here. And so from this, they were able to conclude that it was the humic acid that can degrade the chronic wasting disease. And not only can it degrade those chronic wasting disease prions, sorry, I should say prions here, but that the treatment with humic acid can increase mouse survival. So if mice are injected with the chronic wasting disease prions from, in this case, elk, can see that they succumb relatively quickly. To be quite honest, this is almost 6 months, 180 days post-infection. So this is a very slow acting process where by which the proteins must first cross into the central nervous system and then begin the activity of misfolding some of the other proteins. But with addition of humic acid, they were able to extend the lifespan of those mice that were infected with the chronic wasting disease prions. And there was a dose-dependent effects as they added more humic acid. They could actually rescue up to 60% of these mice from death. It topped out around 2.5% humic acid. 25% did not rescue in much more significant sense. So this is a really big deal because those proteins, as I mentioned, are extremely stable, and this immediately brings to mind ways that we might be able to treat different ecosystems to protect our wildlife. In fact, this was highlighted by the Wildlife Society, a group that is interested in conservation, where the lead author was quoted as saying, given that the prions is shed from infected animals into the environment where it can serve as a source of infection, it's essential that we understand the impact of soil and soil components on this unusual infectious agent. And an unrelated researcher suggested that the next step would be to test the effects of humic acid and addition of humic acid on prion contaminated soils to see if the prions within the soils can be eradicated in the environment. This is a pretty exciting study, and it really complements the recent release of AlphaFold, which is a program that uses artificial intelligence for scientific discovery. This program was highlighted in The Guardian, had a very nice piece that we will link down below, and you can check out if you're interested in predicting protein folding structure. And it would be interesting to see what the protein folding for these prion proteins that have multiple folding compositions would be. All right, we're going to move on to our last story. This is our last story for 2018, and it is a bit of a downer, but so has 2018 been. So I think it's an appropriate way for us to go into the new year with more motivation to treat some of these multi-drug resistant infections, as I'll talk about. So there are two studies, one from Infection Control and Hospital Epidemiology and one from the Lancet Infectious Disease. And they are both looking at the numbers or the effects of antibiotic resistant infections. The one from Infection Control and Hospital Epidemiology is titled Re-Estimating Annual Deaths Due to Multidrug-Resistant Organism Infections. The one from the Lancet Infectious Disease is Attributable Deaths and Disability-Adjusted Life Years Caused by Infections with Antibiotic-Resistant Bacteria in the EU and the European Economic Area in 2015, a population-level modeling analysis. Take-home message from these studies is that more people die from multi-drug resistant infections than previously thought. So, definitely not good news, but not too surprising because the CDC has claimed that the number of people within the United States who die each year from drug resistant infections is 23,000, and this number has been used for a number of years. The true burden is actually unknown, and that's because hospitals do not code specifically for multi-drug resistant organism infections. If someone dies of sepsis, the cause is noted as sepsis and it may be recorded as the type of organism that has caused that sepsis, but it's not known whether that sepsis responded or did not respond to different antibiotics. And so one of the things that I think that this study highlights is the need to track the resistance characteristics and the drug resistant organisms that cause various forms of morbidity and mortality within our healthcare system. So this study, from Infection Control and Hospital Immunity and Epidemiology, is extremely fast. It is a one-page read. There's a lot of quick calculations. I'm going to go through one of them in order to cover this table that gives a new number for deaths due to multi-resistant organism infection. In this case, they took numbers from 2010 with almost 2.5 million deaths in the United States. Uh, of those deaths, 715,000, or nearly a third of those, were inpatient hospital deaths. So we have some sort of hospital code for how those died, right? And of those inpatient hospital deaths, about a third of those, 34.4%, were sepsis related. So somebody who had died from sepsis. And of the different types of organisms that cause sepsis in the United States, the reported rate of multi-drug resistant organisms is almost a third, 28.8%. And so if you do the math here, can come up with 70,837 multi-drug resistant sepsis deaths annually. And this is sepsis alone. This doesn't cover other types of infections such as pneumonia that can also cause very high morbidity and even mortality in people. And so this is where this 70,837 number came from. That's nearly 3 times as much as what the CDC claims on their page. And if you read the rest of the paper, they go through a number of these different deaths due to both infection and multi-drug resistant organisms. There was another paper, this is from earlier in November, that came out from Lancet Infectious Disease. And in this study, it was the first time that the burden of all types of infection with antibiotic resistant bacteria were calculated in terms of disability adjusted life years. So looking not only at death, but whether there was some sort of quality of life inhibition for an X amount of time, potentially extended amount of time, and what that would mean in terms of life years. And so what they were able to find is that, yes, there's a pattern, as you can see in the map on the right-hand side, of the worst or the highest cost in terms of disability adjusted life years in Southern Europe, in Greece and Italy, and it becomes progressively fewer life years as we get up towards Scandinavia and up Norway, Sweden, and Finland there. But across Europe, all age groups are affected. So this is not something that only happens among elderly or not only among certain immunocompromised people. They did find that the burden of these disability adjusted life years was highest among infants. So infants don't have a fully developed immune system. One could imagine that trying to find the correct antibiotic to treat an infection, should the organism be resistant, might have a worse outcome with somebody who has an undeveloped immune system. Overall, for Europe, the summary was 170 disability adjusted life years per 100,000 population life years. And this is close to the total of 183 disability adjusted life years from the combination of influenza, tuberculosis, and HIV infection across Europe. So this is a very serious problem, and I think really highlights the negative effect that these drug resistant organisms can have on societies. So, of course, this is a pretty big deal. One would think that it would have hit all of the major newspapers, but in fact, it was somewhat hidden. It came out over the holidays in the U.S. Thanksgiving, kept people from checking the news as regularly as they do. And in addition to this report, there was also the big news of the climate report, which was released the Friday after Thanksgiving and took up what little science space was available in people's brains at that time. There was a highlight in the Daily Mail, which was a little bit surprising. They highlighted the infection control and hospital epidemiology study that focused on the United States. Kind of interesting for a European or an English tabloid. In the United States, there were only a few mentions of this in trade mags, such as the Center for Infectious Disease. Uh, oh, I forget what the R is, Resistance and Protection or something, an institute at the University of Minnesota. Helio.com, which is an outlet for healthcare providers, also highlighted this study. And here they spoke with the lead author, who said that then a paper came out showing that Spain had approximately 35,000 deaths per year from multi-drug resistant organisms. I knew I had to come up with a better estimate for the US. The US has 7 times the population of Spain, so I knew we had to have more deaths due to MDROs than Spain. All right, that's going to be it for 2018 and Microbial Minutes. Today we talked about speciation and how microbial species do exist, and we're learning more about microbial evolution. We discussed the plague bacterium that existed in and shaped Neolithic life. We talked about how a soil component can degrade hard to denature prion proteins. And we ended with a call for action based on the fact that more people die from multi-drug resistant infections than previously thought. Hopefully, this will inspire you to, use your antibiotics appropriately and to not to get a prescription when you have a viral infection, and perhaps even study clinical microbiology. All right, that's going to do it for today's Microbial Minutes session. If you have any additional comments, feel free to leave a question. We check back quite often. Otherwise, we're off for the holidays, and we'll be back in 2019 with more Microbial Minutes. Don't miss us, subscribe, and you'll get a notification. Thanks, everyone.